For those who weren't logged in earlier, I'm Siri Vaith, Executive Director of CFRI, and I'm just so thrilled to have you here at our conference, and very thrilled to turn this panel over to the wonderful Dr. Julie Desch. So I just want to share a little bit about Julie, Dr. Desch. An adult with CF, Dr. Desch attended Stanford Medical School, where she worked with Dr. Jeffrey Wine in his CF research laboratory. A surgical and skin pathologist, Dr. Desch worked at Kaiser Hospital in San Francisco until she retired. And after working as a wellness coach with people with CF, she completed a mindfulness training, a mindfulness teacher training certification program, and has taught meditation online for the last 5 years, including teaching a mindfulness class through CFRI, for which we are forever grateful. Julie serves on CFRI's board of directors. She is also the chair of our research advisory committee, and we are so thrilled that she will be moderating this wonderful panel on phage therapy. So with that, I will turn it over to you, Julie.
Thank you, Siri, and welcome everyone. Welcome back. Sorry about that little glitch. I want to start off tonight by introducing our panelists. We have Dr. Steffanie Strathdee, Dr. Saima Aslam, Ella Balasa, and Dr. Tom Patterson. So as most of you heard the amazing presentation earlier, Dr. Steffanie Strathdee is a renowned infectious disease epidemiologist. She's Associate Dean of Global Health Sciences and the Harold Simon Distinguished Professor of Medicine at the University of California, San Diego. And there she co-directs the Center for Innovative Phage Applications and Therapeutics, which is the IPATH program she spoke about. After saving her husband's life from a deadly superbug using bacteriophages, Dr. Strathdee and her husband co-authored a book entitled The Perfect Predator, A Scientist's Race to Save Her Husband from a Deadly Superbug. Dr. Saima Aslam is a board-certified infectious disease specialist with expertise in caring for solid organ transplant candidates and recipients, as well as other immunocompromised individuals. Dr. Aslam directs UC San Diego's Health's Solid Organ Transplant Infectious Disease Service, which provides expert care in the prevention and management of infectious diseases in organ transplant donors and recipients. As an associate professor in the Department of Medicine, Dr. Aslam's research interests includes transplant infectious diseases, catheter-related bacteremia, and biofilm-based infection. And then we have Ella Balasa, who is a patient advocate, consultant, and a person living with cystic fibrosis. Since her diagnosis at 18 months old, Ella has experienced countless hospitalization. She now empowers patients and advances research and healthcare strategies through her connections with researchers, pharmaceutical companies, and patient organizations. She's an advocate for the development of novel therapies for the treatment of antibiotic-resistant infections, and through her public speaking, emphasizes the value of the patient perspective. In 2019, she was the recipient of successful phage therapy treatment to fight an antibiotic-resistant infection in her lungs. And we are very grateful that that worked, and she's here with us tonight. And then finally, we have Dr. Thomas Patterson, Stephanie's husband, and distinguished professor of psychiatry at the University of California, San Diego School of Medicine. He's an experimental psychologist with research interests in stress and its impact on health, measurement of functioning and cognitively impaired individuals, and developing interventions focused on HIV prevention in high-risk populations. In March 2016, Dr. Patterson became the first known person in the United States to successfully undergo intravenous bacteriophage therapy we just heard about. And he went on to co-author the book with Steffanie, his wife, The Perfect Predator, detailing the experience. So, welcome everybody. We have, I'm sure, a lot of questions to take and to answer, but I want to begin the discussion by having each of you briefly share your firsthand knowledge and experience of phage therapy. So let's start. We just heard from Steffanie. Let's start with Tom.
Well, thank you for having me.
It's
I guess I would start by saying that I'm extremely fortunate to have married Steffanie. If that isn't obvious to everyone already, planning ahead, obviously, I was very smart in my decision making. But the couple of things that I would mention, and I think I'm preaching to the choir for the most part, but a couple of my experiences, one from the patient side, I'll say, being in a coma, I was in a coma for about a little over 3 months. And I think what's important for people to hear, I don't think this is true for everyone, but certainly in my case, I could hear people talking over me. And that those conversations about futility and my dying fed into hallucinations and a really negative experience on one hand. And on the other hand, having my daughters and Steffanie and friends and family by my bedside talking to me about positive things really gave me a lot of energy to survive and endure what I went through. So I would encourage you to be supportive even when it doesn't appear there's any response from the patient, because it may very well have a profound impact on what's going on. I guess the other thing I would say about being a patient, and this again is something that I know Ella and others who have been hospitalized so many times are well experienced with, but I would say the mindfulness idea, I think, isn't far from exactly what I think made it, for me, survivable for 9 months in the hospital. And that is really, it wasn't taking it one day at a time. It was really taking things 1 minute at a time. It's just not worrying about the past, not worrying about the future, but just focusing on where you are at that moment. And then I guess I just say from a family's perspective, it isn't, as you well know, most of you, it's as difficult or more difficult even for the family. I can't tell you how difficult PTSD was for me, but for Steffanie, my daughters, and others connected with this, it is very, very difficult to deal with the stress of having someone who's near death, and as near death as you could possibly be in my case, and going through the recovery. And once you're out of the hospital, and CF patients certainly know this better than I do, but the recovery is at least as challenging as the actual experience of hospitalization. The rule of thumb is about 5 times as long to recover as you're in the hospital. So in my case, it was supposed to be about 4 years to recover. And I can say, once you've lost all of your muscle mass and have to learn to breathe and to talk and swallow and all of the rest of the functions that you have, it takes longer than that, really. It's a very, very gradual process, and it's as challenging for the caregiver, for the family, as it is for the patient. And with that, I think I'll stop and let the next person speak.
Thank you. Yeah, very well said. I'm just curious, do you remember the hand squeeze moment?
Yes, I do. And, in the book, you'll see that I recall a number of my hallucinations during this period. And at that particular time, I thought I was a snake. And when I say I thought I was a snake, this isn't like a drug hallucination. I was a snake. And I got that idea, I think, from the fact that the tube, when I was intubated, looked like a snake to me, and people were talking to me about I was dying, over me, that I was dying. And so in my hallucination, I was being filmed for a documentary about death and dying. And so when Steffanie squeezed my hand, there was a long pause, she said. And the reason there was a long pause is I didn't have hands as a snake. I had to figure out how to squeeze her hand. So I wrapped my body around her hand and squeezed a little too hard, apparently, but I didn't know my own strength at that time.
That's great. Thank you.
Sure.
I'm sure we're coming back to you. Okay, Ella, tell us about your experience.
Yeah, so I heard about phage therapy, kind of was a coincidence and just I don't know, maybe by the grace of some higher power because it was when the researchers at Yale, Dr. Chan and Dr. Koff were there was a documentary that they were doing with I guess a company that was doing this filming, and it was a patient that was being treated with phage therapy, a cystic fibrosis patient. And I, at that time, was already struggling with resistant infections, was getting intravenous antibiotics multiple times a year. They were taking longer to take effect, not seeing the same bounce back as I had used to before. So I was really thinking about, well, what is going to happen in the next few years? How is this going to be sustained? This need for antibiotics, this level of need for antibiotics. And so this was like winter of 2018, and I got really sick with an infection. I had Pseudomonas on top of, I had like a respiratory bug that I got really sick with. And so I was on intravenous antibiotics, one of them being Clinician. So that's one of the strongest antibiotics that we have available to us. And I, at that point already had been doing a lot of like patient advocacy work and a lot of writing. I had my blog. And so that company that was doing that documentary with Dr. Chan reached out to me and was like, we are doing this documentary. We'd love for you to write just a little bit about what living with CF was like to accompany that video. And that was the first I'd really heard of phage. So they sent me the video first so I could like look at it and kind of base my piece on that. And I immediately was like, I have to try this. And so I reached out. I have a biology background. I was working in a lab at the time. And so I felt comfortable with reaching out to the researchers and talking to them about it and seeing if there was a possibility for me to try it. And so that's what I did. And that was in like December of 18. And pretty much like in the weeks that we were communicating, I was sending my sputum samples to be tested to see if they had phage available that would work for the bacteria that I had. In that period of time, I was just getting sicker. I was not improving with my symptoms despite being on antibiotics. And so in early January of 19, we made the plan that they did have phage available and that I would be treated. And so my doctor, my cystic fibrosis doctor here in Richmond, wasn't aware of phage, had no idea, wasn't very supportive of it. But I felt that at the time, the risk, the benefit outweighed the risk. There was certainly a risk involved. I mean, at the time I had 18% FEV1. So I was on oxygen all the time. I couldn't even breathe to do daily functions. But my mom and I made the trip up to Yale, and I received it through compassionate use, so through expanded access. And I did 1 week of inhaled phage, and I really didn't see much improvement in that week at all, really. So came back home to Richmond, and my doctor here was like, you tried the phage, it's not helping, you need to go into the hospital. So I went down to Duke University where I'm followed for transplant, and they, started me on some new antibiotics, and we're thinking about doing the transplant workup because I was going to get started on that process. And then really within 2 days after that, I immediately started I started clearing the bacteria. So I was coughing up all of the old mucus. The infection was clearing. I could feel it. And I'd used those antibiotics that they put me on at the time many times before. So, and because I was still followed at Yale, they asked me to send sputum samples back to them to see the progress. And so they tested the bacteria, and it had changed resistances to be susceptible to this particular antibiotic that they happened to put me on. And that was Cipro and some other intravenous antibiotics as well. So with that combination, as the phage changed the bacteria, the resistances, then that cleared the infection. And it really was, I mean, it was amazing. It was just everything aligned for me. And that's something I can talk about later too, is just like how difficult it is for, patients to access phage, and how that is, hopefully changing, as Steffanie was talking about, with clinical trials coming about. But, it really was a miracle. And, now I'm an advocate for it, and I have received phage once since then, but hopefully, it's something that I can access again, and many more people can as well. So I'll stop there and I'll let the next person talk.
That was actually more than a phage cocktail. It was an actual cocktail with phage and antibiotics combined in sequence.
That's right. In the synergy, yes.
And are you on Trikafta right now?
I am, yes. So I started taking it about a year after that.
Excellent. Thank you. I didn't mean to mute myself. Let's hear from Saima.
Well, thank you very much, Julie, and thank you for the introduction earlier as well. As you mentioned, I'm an infectious disease physician. I work at UCSD and at the Center for Innovative Phage Applications and Therapeutics. I tend to see patients sort of pre and post-transplant, so people with CF, when they're undergoing evaluation for lung transplant, as well as afterwards, as well as sort of other organs as well. And I'm sure, many of you know this, unfortunately, when people have been sick as often, as frequently as patients with CF, as well as patients undergoing transplant, there's a lot of drug resistance. And we have many patients that unfortunately run out of options, and we don't have good antibiotic choices. And that's sort of really what led me down my path of leading the clinical applications at Center for Innovative Phage Applications and Therapeutics. So the first patient I treated with phage was actually a lung transplant patient. This was our patient number 2 at UCSD in 2017. And since then, we've actually now treated 18 patients with phage for a variety of different indications at UCSD, and have collaborated really with many people, both within the United States as well as internationally, and so been involved sort of as a consultant role with patients, requiring phage or undergoing evaluation for phage, or figuring out if phage really is a good option for a certain kind of infection or a certain, set of circumstances or not. And I'm very interested in really continuing clinical research alongside compassionate use phage, so that, while we're working on developing clinical trials and developing indications that the FDA would allow us to use phage for, we still are able to take care of patients that need it and that potentially can benefit from it. And, we have 2 here on our panel, and there are so many more that I think have benefited from phage, both here as well as globally.
Yes, thank you. And do you have patients calling you, flying to UCSD, or not if they need phage probably, but being transported to UCSD, or is it within the UCSD system that you've treated these 18?
So the 18 are specifically within UCSD, but we've been involved in patients, outside the state. We actually have had a couple of patients that one actually flew in from Wyoming to get care at UCSD. We had somebody else that would drive in from LA for phage, and we have somebody we're currently evaluating in New Mexico. But the goal really is to try and have patients be able to work with their local physicians and get phage there. It's not easy, and certainly I'm sure is a financial issue too, to just get up and fly somewhere to get experimental therapy. So we always try to work with local physicians, the patient's infectious disease doctors, and help develop a plan of how, to treat with phage, sort of a whole management plan, consents, work with them, help them get compassionate use EINDs with the FDA, And so really collaborate with them at every stage of that patient's care, sort of prior to phage, understanding if they would potentially benefit or not, helping them liaise with various collaborators that have phages, for Pseudomonas, which may be Yale or other places, or E. coli, whatever organism it is, to help really build that bridge with basic scientists that have phages, and then help manage the clinical piece of it in terms of developing a treatment plan, how long to treat, how frequent should a dose be, and things like that. So, with Tom's case in the US, and I think in the world, that was the first time intravenous phage had been used. And we were really flying in the dark. Since then, with each patient that we treat, we've learned a little bit more. We certainly have learned how to finesse it a little bit in terms of, what's the best mode of administration? Is it nebulized or intravenous? And part of that really depends on the organism we're treating as well as the indication. So is it pneumonia, like Ella, or abdominal abscess like Tom? Certainly there are many kidney transplant patients with urine infections, et cetera. So trying to be our goal really is to sort of help people regardless of where they are, regardless of whether in the US or not.
Right. And I'm just curious, before we move to Steffanie, have you seen other cases similar to Ella's, where phage therapy would then cause a bacteria to become more susceptible to antibiotic treatment? So you do a combination of the two?
Yes, so we've actually seen that in many instances, in particular for several lung transplant patients with Pseudomonas that we treated either at UCSD or collaborated with other centers such as Cleveland Clinic and others. We've seen that using phage, and this is sort of, I think, one of the benefits of phage, and probably has more applications, that we either eradicate that particular organism that we've sort of super selectively targeted with phage, then allowing other Pseudomonas or other organisms to sort of take that niche, that ecological niche that's then vacated by the MDR pathogen, or at times, such as with the Yale's group, to push the Pseudomonas, so sort of target phage in such a way that the Pseudomonas is forced to become resistant to the phage, but in doing so actually becomes susceptible to a variety of different antibiotics. So when we started off, we've had several patients that have an organism resistant to everything. And then, a week or 2 weeks later, they may have Pseudomonas again, but now suddenly we can use oral ciprofloxacin, which is a game changer. And we've seen that in other organisms as well, such as E. coli in the setting of kidney transplant. And again, going from living on IV antibiotics to then suddenly have an oral option, I think is a big deal. And depending on the kind of organism and infection, at times, you can eradicate it completely with phage too. So I think that we're learning a lot of how the goal is to have the best clinical outcome, and there are different ways of doing that. And I think we're learning that with each patient that we treat.
Yeah, right. All right, thank you. Steffanie, do you have more to add to your amazing presentation for this introductory?
Thank you, Julie. You're too kind. Well, I want to give hats off to the physicians like Saima and to the patients like Ella and of course my husband, for telling their stories because it's hard. It really is hard. But I do think that phage antibiotic synergy is something that we're learning more about. We don't think that phage are ever going to entirely replace antibiotics. They're going to help us use antibiotics in a smarter way and more judiciously without being, just kind of flooding the patient with it. One of the things I'd like to get Saima's take on this, because I've had CF patients ask me, what they could be doing. They don't necessarily need phage therapy now. They're doing pretty well, but is there something that they could do to kind of, anticipate and make phage therapy more potentially better for them if they need it? And one of the things that I've heard some folks say, and I think Saima might agree with me, but we'll see, because I'm not a physician. I'm not going to pretend I'm one either, is that to ask the doctor or the lab to save your cultures, because there's only, like, if you are having a chronic infection with the same type of bacteria, say it's Pseudomonas aeruginosa or Burkholderia cepacia, there's only a certain number of mutations that are going to occur. And maybe it's a lot of them, but it's not infinite. So if you have cultures from the last 5 years that that we as phage researchers, more Saima than me, can draw from those cultures to be able to select phage that are active against the full spectrum of those bacterial mutants. That's what was done for this case in Belgium that I presented in my talk. That they used what's called directed evolution to try to be smart about what kinds of phage could respond to the bacterial mutants that could arise. So anyway, I think I put that out there because, you know, a lot of folks, they give their sputum or like a bronchoscopic culture, and you know, it gets tested and then it gets wasted, like thrown out. I mean, there's something called a freezer, you know, save them. It could be valuable to help us choose better phage for you in the future. Am I off base, Saima, or is that helpful?
So it is helpful, but so what you're describing in terms of, you know, taking bacteria and then exposing them to a variety of different phage and pushing them down different sort of mutations and evolutionary pathways, that can actually be done in a lab setting as well. So yes, we can potentially save isolates. I think the issue is with Pseudomonas in particular, because it's so common, unfortunately, so not each lab has the space or the resources to save, right? So I know for Burkholderia in particular, the CF Foundation, you know, does fund a microbiology lab run by Dr. Lapuma, in which all over the US centers, patients that have Burkholderia, they're sent to Dr. Lapuma's lab, and we can actually go back 5 years or 10 years and get those samples. But this Burkholderia only occurs in about 3% of CF, whereas Pseudomonas is a lot more common. So I think that's helpful, especially now that Trikafta and similar drugs are so common. A lot of patients really are not bringing up sputum anymore. And so that's been tricky in terms of we want to know what's deep down inside, but they've sort of dried out and are not bringing up enough for us to figure out what kind of Pseudomonas they have. So in that setting, something that's been saved previously, I think, is helpful. But the issue with Pseudomonas and CF, we have so many antibiotic pressures over time that keep changing that we really want to know what the patient has now, that we definitely want to get. And sometimes what was there 5 years ago may not be that helpful. But finding out or driving that organism through a variety of different mutations can actually be done in the lab.
Yeah.
And there are certain collaborators that are actually doing that to develop phages that they know will kill not only the bug that they have, but also all potential mutations that can arise. And I think that's sort of the next step of phage therapy, where we're using smart phages, or using, you know, a cocktail that will cover those different eventualities. So if we're able to do that in the lab, we don't necessarily need to go back in time, you know, to collect those previous samples.
Yeah, thanks.
But I think it'll be helpful. Yeah.
Yeah, that's what they did for that Belgian patient. Thanks. I see there's lots of stuff in the chat. So back to you, Julie.
There are a lot of questions, but first of all, I just want to say science is freaking amazing. Devin Wakefield had a really good question that's sort of related to what we were just talking about. He says there's been some research, a lot of research, to indicate that in many CF patients, sputum samples are not very helpful to predict how well a patient will respond to antibiotics and which antibiotics to choose. Will phage have this issue as well? Are there better ways to choose phage/antibiotics than sputum cultures?
So I think at the moment we're limited to sputum cultures. And I think that has the same issue as antibiotics in terms of, are we really getting the organism that is causing, you know, the lower respiratory tract infection? But again, as we just mentioned earlier, there really is a push to try to develop sort of these evolved Pseudomonas cocktails that should get at a variety of different Pseudomonas mutants. And I think, you know, I think as we move forward, that probably will be a better way rather than just what we have in the sputum today, because that may not be what they had a week ago and it may not be what they have next week. But at the moment, that's where we're limited. And maybe Ella or Steph or Tom would add in.
I keep forgetting I'm muted. Tom or Ella, do you have anything to add? We have another
Well, I
question.
guess the only thing I would say, you know, and I don't know if Steph mentioned it in this particular talk, but the idea that, you know, eventually we're going to have this library of bacteria, and against that, we're going to have a phage library. And then, you know, I mean, using probably, you know, mathematics and whatnot, you'll be able to predict which one or combinations will work. But that's kind of in the future. That's what, you know, I see as the, you know, the next generation of what we're doing with phages and what's really needed. Because right now, phages are scattered around the world. I don't mean that in a sense of, yes, they're all over us and they're in the sewer and everywhere else. I mean, the libraries that exist and the labs that have them have a limited number and they're scattered all over, and we have to go to every place to find them each time. That's not efficient. It needs to be a library that's available to everyone in the US and globally, you know, and there should be a library in Africa, there should be a library in Europe, there should be one in Asia, there should be one here, and they should be contributing to each other. And I think that is what's going to really move this forward.
And quickly to add to that too, I mean, that coincides with, you know, that time issue, right? As the bacteria colonies can change so rapidly in the lung and in CF and what other, you know, other infections, that you know, to having this phage library too, like, and understanding what phages are out there to treat different strains of bacteria can make that process much quicker to treatment. But once you isolate the bacteria, and then you can very quickly move into treating actively with an effective phage, rather than, you know, isolating the bacteria, finding the phage, growing it, purifying it, which takes a long time. And then at that point, that phage, I mean, that bacteria could already change its susceptibilities and the population. So that's another point to that.
Yeah, just to chime in, because there's a couple of related questions in the chat about this. That, you know, if you have a phage library that's ever expanding, because, you know, bacteria are evolving, right? So it isn't like a static phage library that you make it once and then you never need to update it. You need to update it all the time with the bacteria and the bacteriophage. You could go back to that like phage library and use it to identify new phage if a bacterial mutant becomes resistant to the first phage cocktail. In fact, that's what was done in Tom's case. Just opportunistically, we were able to identify, you know, a new phage that was going to be active against the bacterial mutant, and it turns out it was synergistic with an antibiotic, so it was a double whammy, which is terrific. There's also a question in the chat about how quickly can you identify phage matches and a phage hunt for someone. It really all depends, and that goes back to the phage library too. If you have a really expansive phage library, you might be able to find matches right away, right? I mean, the Navy's phage library that was used to phage hunt for Tom, they found phages that, you know, very quickly. They'd never used them on humans before. It was just a bunch of rats and rabbits, so it was still really experimental. But these days, you know, like that's the ideal, so that you don't have to go and collect, like, you know, swamp water or something like that to find phages. But if we can't find any phages that are already characterized in a phage library or in someone's research lab or a company that's willing to give them, then we do have to go to those sources, and that takes longer. So when people ask us, you know, how long does it take? It really does. It depends on the organism. For Pseudomonas, we have pretty good phage libraries. For MRSA, we're getting better phage libraries. For Klebsiella and E. coli, pretty good phage libraries. For Burkholderia cepacia, thanks to the CF Foundation and Saima's, you know, research with her partners around the world, we've got better, you know, phages. But again, they're phages that sometimes need to be manipulated to be optimized for phage therapy. So I think that what we're going to see in the future is more phage that's genetically modified, like the case of Isabel, or even synthetic phages might be needed, where it's like, almost like a 3D printing kind of approach that some folks have put forward, where you have the sequence of the bacterial DNA and you generate phage to match, and that can be done. So the field is rapidly evolving, and so I'll just stop there.
Great, thanks. Yeah, I was going to ask about that. As the research advisory committee member, I have seen a lot of proposals coming in from San Diego about, building phages, essentially, manufacturing phages. And I just wanted to get your input, like how far along do you see that? How distant in the future do you anticipate that is going to be? Because it seems like that would be like the perfect solution, we can build it.
Yeah. Do you mean building phage from scratch, not manufacturing for clinical use, right?
Building phage, yeah.
Like synthetic phage.
Synthetic phage, yeah.
There
Like
are companies.
For instance, there's a process where you just use the tail of the phage to, connect with the receptor, and then, you kill the bacteria that way. So I don't know, maybe that's out of your field, or
It's being done. Yeah. I mean, I don't manufacture them, but I know that there are groups that have built synthetic phages, including several companies in Europe as well as the US. And the goal is to try and use the synthetic phage for treatment of specific infections as well. And some of the synthetic phages actually carry payloads inside them, which could potentially be an antibiotic that, for example, and I'm just going to give you an example, but Pseudomonas, when it becomes resistant to meropenem, one of the ways of that it becomes resistant is that it pops out the antibiotic. So there are ways of actually adding payloads within the phage that then puts it into the bacterial cell that then can antagonize development of that resistance mechanism, for example, or other ways in which you can directly kill organisms. There's interest in using synthetic phages and actually trying to kill cancer cells. So outside infectious disease, there certainly is a lot of application for that as well, and interest.
Yeah, that's very interesting. Okay, so a question that has been drawn up here is, can you talk a little bit about resistance to phage therapy? Like, how does that come about, and what do you do in that situation?
Yeah, I can start off with that, and others are welcome to chime in as well. So what happens is when we have phage, and I think Steffanie showed this, right? So when we have phage, they basically insert their DNA or RNA into the bacterial cell that then takes over that cell's replication, right? And starts making lots of little virions. And when they basically kill the bacterial cell to release themselves and attach to more bacterial cells. So resistance mechanisms in bacteria can occur at a variety of these steps. So bacteria can get rid of the receptor that a particular phage uses to attach to itself. So that if a bacterial receptor is gone, the phage can no longer attach to that bacterial cell and kill it. There are also ways in which bacteria can develop enzymes or block various enzymes within that replication pathway so that a virion may enter, DNA or RNA material may enter the cell, but then it's a dead end. It's an abortive infection, for example. So there are a variety of multiple steps along the way that bacteria can mutate and develop resistance to save themselves. And also at times, Pseudomonas, again, is an example in which certain phages can infect a Pseudomonas and live in it, what we call lysogenic phages. So they live in harmony, if you have it, with the bacterial cell. But then they'll release or cause mutations or release proteins so that other phages cannot attack that same cell and kill it. So one phage is in the Pseudomonas already, is living happily, but it'll prevent other phages from coming in and killing it. So I think there are a variety of different mechanisms in which that resistance can occur. And so in terms of overcoming it, and we've done this in several patients, we've used sequential phage combinations. So we kill, start off with killing one group of Pseudomonas. And this is again an example. As they develop resistance, you get other phages that kill the resistant mutant. So we've done that successfully. Or you can develop, sort of a super cocktail that will kill all, potential mutations as they arise, so that we don't have to worry about it. So I think there are a variety of different ways of trying to make that happen. Sometimes development of resistance to phage is not a bad thing because it may make the bacteria much weaker. It may be that when they become resistant to phage, suddenly now we can kill them with Cipro or with an inhaled antibiotic. So, and that too can be used to our clinical benefit. And these are all sort of predictive pathways that we're now able to sort of harness for patients' best clinical outcome.
Has there been like sort of machine learning mechanisms or artificial intelligence to kind of figure out the evolutionary pathways of a phage and a bacteria and how to combine all of this with antibiotics? Like, is that?
That's actually being done. So there are companies and research labs that are working on machine learning algorithms to figure out which phages would go best together in a cocktail. And how many phages should go in together in a cocktail. And it could differ depending on, the type of bacterial infection. And I see, in the chat, there's a question about how these genetically modified phages are being done. Well, the one that was used on Isabel, this is a precursor to the CRISPR-Cas gene editing, which is now becoming more common. They used a technique called recombinating, where they clipped out the repressor gene that makes that, phage kind of the sleepy kind that integrates into the bacterial cell DNA. And clipping that gene forced it to become lytic, the phage rage kind. So that's just one technique. But now that we have CRISPR-Cas gene editing, we could study what aspect, what lifestyle of the phage, where do we need to kind of make it better? Just like what Dr. Aslam was saying, that, there's the attaching, the phage enters the cell, it starts replicating. There's ways that we could make it better killers, better bacterial killers. And so we're just learning about this. I mean, when I say we, I mean the whole field, because the field is developing very rapidly. And it's a field where CRISPR-Cas gene editing and phage therapy are coming together. And there's a bunch of different companies, like one company that I don't have any relationship with, so I can mention, Locus Biosciences has received like an $800 million investment from Johnson & Johnson. So pharmas are paying attention. Yes, we do need to incentivize, the industry to kind of get involved to bring things to scale. It's a delicate balance right now, but we do need more basic science research to be able to inform the routes that we need to go to make phage therapy better.
How is our country doing in comparison to other countries with this science? Like, are there other countries that are more advanced, or are we kind of cutting edge and they're looking to us?
Saima probably knows the best of this because she's been working with colleagues in Israel and Canada. So I'll let her chime in unless others have something to say.
There's some countries that certainly have more years of experience than the US, in particular Eastern Europe. In terms of cutting edge, I feel like it's the global community. So there are many companies in Europe, there's centers in Australia, there's centers in India. I have collaborators in Israel, and I feel like there's no sort of this country is better than that country. We have different sort of years of experience, but I think in terms of what's cutting edge, you could be anywhere in the world and be able to get it. So I don't, I guess, find us really behind or in front of anybody.
Okay, cool. Okay, so I'm going to go to I'm going to try really hard to figure out what's already been answered here. Quickly, valley fever. Has there been a phage developed?
Yeah, valley fever, unfortunately, is caused by a fungus. So bacteriophage therapy, or these are viruses that attack bacteria, so that wouldn't work. I mean, there are mycophages that are, viruses that attack fungi, but those are even less studied than, than bacteriophage. I noticed that there was a question about adverse responses to phage in humans, and we haven't seen any. Yes, antibodies can be developed against phage, and resistance, like bacterial mutants, can be arise due to phage, but we haven't seen, like, we injected 1 billion phages per dose every 2 hours into Tom Patterson. That was our test case. My husband, as he likes to say, because he's a psychologist by training, became the large animal model. He was the experiment. And so since then, we have literally used intravenous phage therapy. When I say we, this is mostly Saima. In almost every case, we've gone intravenous because we haven't seen adverse effects. So the FDA is very comfortable with intravenous phage therapy. So that's really good news.
I'm just going to add a little to that. So what happens is when we get a clinical preparation of phage, it is grown within a host bacteria. And so the clinical preparation can have endotoxin from the bacteria. So there is concern that perhaps when we inject, that can cause a reaction. However, all clinical phage preparations are very carefully cleaned of that endotoxin, so there's minimal. And also we make sure they're sterile prior to giving to patients. And so those are sort of the two main concerns in terms of contamination. So generally it's developed in a way to sort of clean it of contaminants and of endotoxin. There is a few cases, case series, one in particular published from Australia of patients with Staph aureus, bacteremia, and endocarditis. In some of those patients, they did note an LFT or liver function test abnormality. But it was unclear if it was due to the phage or was due to the patient's critical illness. So it is something we monitor carefully when we treat patients. So we'll monitor their kidney function, their liver function, their blood cell counts, as well as inflammatory markers. There are studies showing inflammatory markers can, in some patients, increase after the intravenous phage, but clinically, it hasn't led to sort of a clinical manifestation of that. I've had one patient that did develop shortness of breath and wheezing after a very particularly high dose of intravenous phage. But when we diluted and gave the same phage in 1 log less dose, they tolerated it just fine. So the concern was potentially, there may have been a contaminant within that preparation. But I think as we move towards GMP or good manufacturing practice level development of phage preparations, that should be less of a concern. But every patient we treat, we do monitor them very closely for any adverse event. And in particular, these are the things we look for. And I'm going to add in one more thing related to that. Because patients, and depending on the disease state that we're treating, in particular, the example I want to give is for a few patients that had cardiac device infections. So these are large ventricular cyst devices that were infected for several years with Pseudomonas. They have a variety of Pseudomonas isolates sort of on that device. And what we culture in the blood is sort of tip of that iceberg. And, that's what we focus the phage against. Then we have had actually 2 patients I've had, and I think there's been at least one at other center, in which a week after starting phage, a patient did develop bacteremia. And I think it's just because we're killing that biofilm or that slime layer so effectively that we're seeing it in the blood. So that's something we watch out for with these cardiac device infections as well, that just, looking for bacteria in the blood, they were susceptible to phage. It's just that they had such a massive, again, bacterial burden that all got released. So we do watch out for these things. But in general, there's been no sort of serious adverse effect, that could not be clinically managed. That hasn't happened.
I can add just a personal note on the, the inflammation or like the immune system response. And I've done inhaled phage, and I did experience after inhaling, a couple of hours later, I did feel some shortness of breath, some tightness, like airway tightness. But it was remedied by doing some albuterol, which I do, 3 to 4 times a day. So, as long as I was keeping up with that treatment as well, the symptoms were not severe or, not unmanageable. But that is something that I would experience when I did it.
Ella, Devin wants to know what phage tastes like.
Tasteless. It's diluted. At least what I used, it was diluted in like normal saline. So, not even like a hypertonic saline, not that strong of a salt taste. Just
Well, I still like to tell my husband that he's full of shit, though. Like,
Caitlin Rogers says, realistically, what percentage of the world's population can be treated in the future? At the moment, it seems like it's a pretty cumbersome process.
well, I mean, I guess this is a global health question, so I'll try to take it on. I don't think we really know at this point. I mean, we obviously have logistical hurdles in terms of bringing phage therapy to scale. But I think if we had a phage library at the scale that I've been talking about, that, the escape pathogens were each one of those letters, ESKAPE, stands for a different superbug of major worldwide health importance. We had that plus, some of the other, infections that affect CF patients like Burkholderia and Stenotrophomonas and some others. We could really make a dent because phage are sourced so easily. And the most complicated and expensive part of it is the purification and the expansion of it in a GMP facility if we're going to treat people that have less severe illnesses. So I think it's doable. Could we really obviously need to get ahead of the superbug crisis? It's outside of the CF community. It's still a huge problem. So it's something that everybody should be concerned about. I also saw in the chat that somebody asked, is it possible to synthesize phage, like make a new strain specifically for a specific bacteria? Yes, there are synthetic phage companies and research labs that are working on this as well.
Thanks. Francine Bion wants to know if there is phage available for Achromobacter thylacosidans. Is that a potential target?
There are a couple of labs that we know of that have one in particular at San Diego State University that has a good Achromobacter library. Again, that's a research university. They don't have the ability to do the manufacturing side of it, but if they've often agreed to do phage hunts when we get those requests. And if they find that they've found several hits with phages, then we often have to turn to another lab to grow those phages up to get them ready to give to people. And that's why we also need GMP facilities. And those are surmountable barriers. That's just money. That's just up to people like me to, find it.
Mm-hmm. There's a few different times people have been asking about the purification of this. I think people are freaked out by, these phages are accessed in swamps. And so like the purification process is apparently something people want to know about.
So phage may be picked up in swamps or other places, but what we end up with is basically a Petri dish with pure bacteria. And that pure bacteria is then inoculated in a growth bacteria in sort of a big jug of fluid and grown in it. And so once you have the final product, it actually runs through a variety of different filters, a sort of a hemodialysis type machine that also cleans everything out. So in the end, you really are left with phage within their medium, or whether it's normal saline, or D5W or something of that sort. There's a pretty detailed sterility testing done of that product prior to giving it to any patient. And again, we actually test out the actual concentration of endotoxin within that product, which again is minimal to close to zero prior to giving it to a patient. So I don't know, Steffanie, if you showed pictures of the final product, but usually it's clear fluid.
Yeah, and I guess also people don't realize when they take an antibiotic like, penicillin, cephalosporin, whatever. I mean, it looks like a nice little pill, but penicillin came from a mold. That's a green, hairy mold, like on a Petri dish. So, I mean, obviously you're not like eating that mold, right? You're eating a purified, manufactured, piece of that. And that's what, when I say GMP, it's GM as in Mary, P as in Paul facility, because I saw someone, I was asking what is that? That stands for good manufacturing practices. I mean, we feel like we need to treat phage like a living antibiotic. So it needs to be put through the same steps that if you were developing a new class of antibiotics from scratch, you'd need to put it through the same kinds of tests. And so a facility that, has the standards that are rigorous to be able to do that's what I'm talking about. And those are, it's all, another name for that is like a bioreactor.
Doug Modlin has a good question. Has there been, wait, where'd it go? Oh, never mind. His question was answered. Okay, so we have another one that's interesting. Ella, question for you. You mentioned having a second phage treatment. Can you talk more about why you had to go back? Were you seeing more antibiotic resistance again? How did that happen?
Yeah, so I received phage again. So I received it in January of 19 and then again in May of 19. And then actually most recently too in the last couple of months. But because the CF lung is, it's colonized with bacteria. So, the phage acts like an antibiotic, right? Like we take antibiotics, we have to take them many times because it doesn't fully eradicate the bacteria. There's still underlying infection. So, same thing. So phage has to be dosed again and again. I mean, obviously, maybe Saima can speak to this more, but maybe a longer term course of phage where, maybe, I don't know, a couple months treatment or something that could really beat back the infection you could potentially see eradication of the infection from that point. But, just like these short treatment courses, at least what I've received, it just beats back that acute infection, but it does come back. And so then that's why I received phage again. And, even now, I mean, I do use antibiotics pretty frequently. Even with being on Trikafta, I do a lot of inhaled and oral antibiotic courses. And I did actually receive another round of phage just earlier this year, and it wasn't as effective this time. And I think that had something to do with the fact that it took a long time between the time that my sputum was sampled, which it was probably like 3 months difference between the time that my sputum was sampled and then the phage was purified and sent back to me, and then I inhaled it and did it intravenous. So, that's another barrier and I think a potential issue that can arise. But yes, so that's the answer then as far as why I needed phage again and why it has to be dosed again and why I still have infection.
Thanks. Okay, we have, go
Sorry,
ahead.
I just want to add a little bit to that. What I wanted to say was, so the duration of therapy is something we're learning about as we treat different indications for treatment. And so how long to treat, I think, is different based on what we're treating. So, and lungs in particular are never sterile. And that's sort of what I tell all my, I think we know that. And so usually if we're eradicating one bug, something else is going to come into that niche. And I think the goal is to develop a cocktail that whatever comes into that empty niche that's vacated by the MDR organism is something that we can then treat later. Because the issue with CF, the bacteria is sort of part of the problem or is the symptom of the underlying illness. And that's sort of what Trikafta is trying to solve is, to help fix that problem at its base. So I think I think for lung infection, it's probably not realistic to think that we will eradicate all Pseudomonas in its entirety forever, because it never, it's, you know, it's always in constant communication with the environment. And so something will come and take its place. But the role is, in my view, and I think different researchers think about it differently, the end game is for somebody to have good lung function. And so for some people, the bacteria may just be living there, but is not really causing that lung deterioration. And so fixing or getting rid of the Pseudomonas may not improve their lung function. So I think that's again where it's important to understand who may benefit ahead of time, so that, you know, we pick the right patient. And I think that's what one of the current clinical trials will try and figure out too. So again, I think we're in a very early learning phase at this time.
Okay, cool. We have time for one more question, and Paul Quinton has a really good one. How and how long can phage be stored before use?
Well, you know, the phage that was used to treat Tom had been sitting in a flask on a lab, like, you know, desktop for a couple of years. Some of the other phage that was used, like, have been in a freezer, you know, or it, like basically phage are inert. You know, there's even a debate as to whether viruses, which phage are viruses, are living or dead. They just kind of are in this suspended animation. In the book, I kind of say like a coma, you know, until they come into contact with their host. So they're just kind of sitting there waiting, right? They're half DNA or RNA and half protein, and they come alive, so to speak, when they bump up against their bacterial cell host and go, aha, yum, 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 yum. So basically, like, you know, there are ways that you can denature them, like if you spin them around in a centrifuge or you put them in a lot of heat. But, you know, in general, many phage are able to just sit there and wait for eternity.
Wow. Field of pure potential. Y'all, this has been amazing. We have to end it because it's been over an hour. But thank you very, very much for your participation. There's so many questions we didn't get to, but we really, really appreciate you coming here and speaking to us about this.